বা এটা থাকলেও হবে।
এটা থাকবে, ওকে।
হ্যাঁ হ্যাঁ।
Dashboard, operational dashboard, and so many more right from the
holiday planning मतलब tapi itu
Okay. স্ক্রিন শেয়ারিংটা অফ করলে ভালো হতো।
স্ক্রিন শেয়ারিংটা অফ করব?
হ্যাঁ করতে পারে, হ্যাঁ করে দিন তাহলে।
এই আমি ইন্ট্রোডিউস করবো যখন, তখন আমি একবার বলে দেবো যে প্লিজ শেয়ার ইউর স্ক্রিন, তখন স্ক্রিন
হ্যাঁ,
শেয়ার।
আচ্ছা, স্ক্রিন শেয়ারটা যেন ওই মোড
নিচে
থেকে
থেকে
পাবো?
নিচে একদম নিচে দেখুন গ্রিন কালারের একটা স্ক্রিন শেয়ার অপশন আছে বটমে, যেখানে সব সমস্ত রকম ইয়েগুলো আসছে। আচ্ছা আপনি ছেড়ে দিন, দরকার নেই মলদা, দরকার নেই, কারণ ওটা করতে গিয়ে আমাদের তখন টাইম লাগে।
হ্যাঁ
আমি
আচ্ছা, স্টপ শেয়ার, একটা স্টপ শেয়ার আছে, এটা কি ক্লিক করে দেবো?
হ্যাঁ তাও হবে, তাও হবে।
ওকে।
খুব নয়েজ আসছে সময়, কি একটু আগে থেকে প্ল্যান করে করতে পারলে না?
আমি দরজা লাগালেই তো দরজা পিটাতে লাগে।
ঠিক আছে, আমরা লাইভ আছি, এত গল্প করে লাভ নেই। ওকে, স্টার্ট করছি। ওয়েলকাম এভরিওয়ান, thanks for the joining. This is our free webinar in this series, and we are very lucky that today we have another expertise. We have a different field of experts, which is in bamboo crop. He is a very reputed and for a long time he doing the research on bamboos. Dr. Malay from Presidency University, Kolkata. Today we are organized in the same pattern. First we are go for the presentations, and then that time we also collect our questions answer from the YouTube. Then we go for the interview sessions, and finally for the question answer round. Or if your questions are enough, then we go for the after presentation, then question answer round. And at the end of this webinar, during the I am sharing a link for the certificate applications, and those are already registered, so they are able to apply through these links. After scrutinize the link, their informations, I will issue the certificate after 2 days. It's in 22, it will be available on our website for the download. I will request Soma to introduce Bioingene and speakers, and once again Dr. Malay for joining Bioingene webinar series. Thanks, Soma.
A very warm welcome to all our viewers of Bioingene, a platform dedicated to the promotion of plant science research. The esteemed guest speakers on our platform are mostly plant scientists who present their research, and future scientists from around the world gain knowledge, perspective, and inspiration. We do this through our webinar series, interview sessions, and publications. Thank you for being a part of today's webinar. As more people are joining in, let me provide some housekeeping information related to today's talk. Please note that after attending today's talk, you can apply for a certificate of participation. For this, you need to submit the feedback form that will be provided multiple times in the YouTube chat after the presentation. When you fill out the feedback form, please use the same email address you used in the registration form, and remember to mention your full institute name and address. Mismatch in email ID may result in non-identification of participants, and your certificate may not be issued. You can collect your participation certificates after 2 to 3 days from our website. Bioingene does not send certificates through email. Please make sure that you have enabled YouTube chat on your device so that you can interact and submit your webinar related questions. We will collect all the relevant questions for our speaker. Our esteemed guest speaker today is Dr. Malay, Assistant Professor and former Head, Department of Life Sciences, Presidency University, Kolkata, India. He will be speaking on the topic Genetics, Genomics, and Breeding of Tree Bamboos, Emerging Prospects and Future Challenges. Dr. Malay has years of teaching and research experience. His areas of interest are plant biotechnology, genetics, and genomics. He has held positions as the Alexander von Humboldt Fellow, Institute of Biochemical Plant Pathology, Helmholtz-Zentrum, Munich, Germany, from 2009 to 2012. As NSF Postdoc Fellow, Institute of Biochemical Plant Pathology, Virginia Tech University, USA. As National Research Council Associate, U.S. Environmental Protection Agency, Western Ecology Division, Oregon, USA. He has written many book chapters and has research publications in high-impact journals such as Plant Physiology, Current Biology, Molecular Biology and Evolution, BMC Genomics and Frontiers in Plant Sciences, Scientific Reports, Nature Group, and Biomass and Bioenergy. He is the editor of international journals like BMC Genomics, Frontiers in Plant Science, and Advances in Bamboo Science. He has served as reviewer in many international journals like PLOS ONE, BMC, Scientific Reports, to name a few. He is a much beloved professor by his students, and I'm positive that all the students and scholars who are viewing today's webinar will learn a lot from his expertise. I request Dr. Das to please share his screen, and let's begin today's talk.
Thank you very much, Dr. Mojumdar and Shuma Madam. I hope I'm audible, right?
Yes, everything is okay.
Okay. So I will go ahead.
In the bottom, there is a screen share, green color options. Just
share.
Share.
Okay. So is it visible now?
No, it's coming. It's now it's okay.
So I'll go to the
yes, full screen mode.
Full screen mode and presentation mode. First of all, thank you, Dr. Mojumdar, for this invitation. I'm very excited to be here today in Bioingene. I know it's a very vibrant platform, and the aim of this Bioingene is to promote plant science research and teaching, primarily aimed at the students and research scholars. So today I'll be talking about a plant group, as you see, is not a model plant. Normally, we always talk about a plant. Excuse me, is there a...
No, it's okay.
It's fine, no?
Yes, it's fine.
Okay. So normally people talk about the model plant like Arabidopsis or rice. And there are many reasons why people prefer to restrict to model plant. For instance, if you just think about Arabidopsis, there are many reasons why people want to restrict themselves to Arabidopsis, and there are 100 reasons you can come up with. Here, I just summarize a few of them. For instance, for this plant, the life cycle is very short. Within 2 months, 2 to 3 months, you can complete the life cycle. It has a relatively decent number of chromosome, relatively lower genome size, which makes the geneticists to do genetics much easier. The growth stages are quite defined, and it is so defined that we know that exactly in a particular growth condition at a particular time, what would be the exact number of the leaves, the rosette sizes, and all these things. And more importantly, it is possible to manipulate the growth system. For instance, if you want to promote vegetative growth, you can grow Arabidopsis under a shorter day, while if you want to accelerate the flowering time, you can grow it under longer day. So it gives you much more leverage, flexibility to grow the plants in your own way, and also a very easy transformation system has already been developed. And also for majority of the genes, the mutants are available. So that is why, a lot of progress has been made so far in terms of our understanding about how plant grows, how plant responds to its environment, and how different genes, the genetic pathways, the proteins, the metabolites, and their interaction, how together and they shape the growth and response of Arabidopsis. But the problem is, life is not about model plant, because if you look around you, you will see majority of the plants that have commercial importance, they are not model plants. I don't know if the slide is changing.
Yes, we also.
Just let me see why.
And there is two other window also. These are why these two
I
kind.
think, just excuse me. Somehow, yes. So now I think it is okay. So is the slide is visible? Yeah.
Now, okay.
Okay.
Changing.
So the commercial. The problem is Arabidopsis is a nice model system with all these unique and interesting features, but the problem is it does not come up with much of the commercial values. We do not produce food, fiber, any kind of commercial importance that can be generated from Arabidopsis. That is why for the plant scientists, for a long time, while life was much easier with Arabidopsis, but after some time, they were forced to move beyond the model plant. And I'm particularly interested about trees. So this is one kind of extreme of the non-model plant, which is not easy to grow in the lab, which has the perennial life cycle, which has a very long life cycle, let's say from 20 to 30 to 100 years, which has a very complicated genome. And as a result, it can pose a lot of technological problem while you are doing many different experiments. And here I put two trees. One is poplar. Poplar is one of the model tree for the plant biologist. But we are not even aiming for this model tree. We are aiming for another tree-like plant, and we call it kind of like an orphan genome, because we believe that not much attention has so far been given to this plant group, bamboo, although it has a unique potential to offer many interesting applied and fundamental aspects of plant biological information, and some of the experience that we had gathered over the last 20 years or so that I'll be sharing with you today. So that is my plant group, bamboo. And as you see in this cartoon, it's a tree-like habit, it's perennial, the ploidy level varies starting from diploid to hexaploid. The life cycle can be extended up to 120 years. The chromosome number is quite variable, starting from 22 to 72. The genome can be as big as 2 GB, and the gene number is again variable within from 24 to 47,000. So that already gives you a clue that the biology of bamboo is going to be complicated in terms of doing genetics, and some of that thing that we are trying to do. Quite often we are indeed facing problem, but at the end we are getting some rewarding results that I want to share with you today. So this is kind of like the majority of the tree genome, where most of the emphasis has been given so far. As you know, like Pinus, Picea, Populus, Eucalyptus, Quercus, and majority of them have been given emphasis because of their commercial importance, primarily due to their timber yielding property. But we are actually aiming on bamboo, which we call, as I already told, is a neglected tree genome, but it has a huge commercial impact. This data is quite old, around 2017, but as you see that in terms of both export and import in all different countries, we are talking about millions of dollars, and that graph is drastically increasing with time. So that is why it is very important now to understand the biology of this plant. And why it is important? Because in countries, majority of the countries like Asian countries or South American countries, bamboo can be considered as a kind of regular day lifestyle, part of lifestyle of us, because they can be used for many different purposes, such as food, fodder, fiber, or so many different purposes. Even United Nations, if you just consider the 17 Sustainable Developmental Goals, this is one of the recent paper that we recently published in collaboration with our Chinese colleagues, and where it was strongly argued that bamboo has a important potential to kind of cover 7 out of the 17 sustainable developmental goals that has been set by United Nations. So it is not only about the commercial aspect, but also about the sustainable development and green development. Bamboo has a enormous potential to contribute in future. In terms of species diversity, the world is fortunate enough to have a lot of species diversity. Here, as I come from India, I show here the data from India, and as you see that India is blessed with a huge amount of bamboo species diversity, and the majority of them are particularly restricted to the northeastern part of India. So huge biodiversity means huge genetic repositories, and a systematic assessment of that genetic repository can be able to give us some genes that can be utilized for many different commercial purposes, and some of the examples from our own work I'll be showing you today. Now, before you start doing genetics, any crop that you're working on, first you have to have a fair bit idea about the genome of that plant. And as I already told you, that bamboo has a very complicated history of polyploid genome. As you see here, that it has like all these different kind of genomic composition, that is A, B, C, D, E, and H, and they kind of cross with each other and form different kind of allohexaploidy and produce these four major lineages. One is ordinary, which is composed of AABB. Then bamboo C is composed of CD. Bamboo C is composed of C, D, and E, and while Olaida is composed of H genome. And in terms of the genome sequencing status, a majority of the sequencing efforts have been contributed by the Chinese scientists, and Phyllostachys heterocycla has been the first bamboo genome which has been sequenced, and this is kind of at the draft genome stage, and for genomic studies this is being used as a model bamboo genome. But 4 more genome has eventually been sequenced, which are Olaida, Radia, Guadua, and Bonia, and emergence of these genome sequences actually giving us leverage to perform evolutionary biologic studies in comparison to related allied monocot genome such as corn, rice, wheat, or brachypodium. Now, so far, just in a nutshell, I want to tell you that if I want to summarize, that the major areas where a lot of work has been done in bamboo genetics, genomics, and breeding, these are the major areas such as in terms of development of molecular markers with the purpose of authentic identification, standardization of the tissue culture method, and in some cases, the transformation protocol standardization. A lot of work has been done, including our group, in the flower, pollination biology, and breeding, because bamboo flowering offers many interesting biological questions to answer. This is a new addition to this big list, and we are contributing to this list actually because bamboo has the potential to evolve as one major contributor in the bioenergy sector. And then pest and disease management, post-harvest management, genetic diversity, phylogenetic analysis, all these fundamental areas also, quite some progress has been made by the scientists. So for today's talk, I'll be both showing you some example of how this kind of orphan plant group can be systematically studied in order to find its application or its utility both in the applied science as well as to understand the fundamental biological concept of how a plant grows and what are the genetic circuits that control that growth. So let's start with the applied aspect, and I want to discuss with you that how bamboo has the potential or can be used for the bioenergy sector. Now, as you recall, that one of the major problem of today's world is the security of energy, because you all will kind of agree that these energy resources, which is kind of so far has been dependent on non-renewable sources like petroleum, coal, natural gas, and nuclear, and so on. And our life has been quite happy for several decades with this kind of non-renewable energy sources. But we got to be careful, because this resource is going to be abolished very soon. So that is why we have to look for alternative sources, which is the renewable sources. And so far, in terms of renewable sources, there are alternative sources like solar, wind, and hydropower, where a lot of investments and energy are being kind of invested. Now, being plant scientists, we can focus on this plant biomass biology or lignocellulose biology and LCB, and we can think that how lignocellulose, or enormous amount of carbon that is being sequestered by the plant by the process of photosynthesis, can be converted back to energy, which can be used in our daily life. So that is the lignocellulose biology, LCB, and that LCB, if we think about the plant, can be again of three different sources. One is like from edible sources, one is non-edible sources, and another one is algal biomass. Now, edible source, as you know, is not a viable option, because if we use food to use energy, then the cost of food will go up. Similarly, algal biomass is an emerging area where many people are investing, but in parallel, plant scientists like us, we are focusing on non-edible plant sources if this can be used as a bioenergy. Now, as I told you, the LCB is usually stored in the cell wall. So that is why first you have to dissect the cell wall to understand the biochemistry of the cell wall. What's special about cell wall so that it can be used for bioenergy production. Now, as you know, that cell wall is primarily made of several layers, which are like primary cell wall, secondary cell wall, plasma membrane, like that. But biochemically, it is mostly composed of polymers like cellulose, hemicellulose, and lignin. Now, for energy purposes, we want to get the cellulose out of lignin and hemicellulose matrix. Now, the cellulose usually in the cell wall is embedded in the matrix of lignin and hemicellulose. So that is why the first challenge of this kind of a bioenergy research is to use some kind of biochemical or physical treatments, which we call pre-treatment, or a combination of both, to get the cellulose out of lignin and hemicellulose, or in other words, to get the lignin or hemicellulose out of cellulose. And once you get the cellulose, then that cellulose, by saccharification reaction, you can break it into the simpler glucose unit, and that glucose can be used as a substrate by fermentation process to produce the bioethanol. So that is the whole kind of background of how a cell wall based lignocellulose biomass can be used for bioenergy. Now, this is the situation and pledge that the Indian Honorable Prime Minister has made, that by 2047, the target for us, that is for India, to becoming energy independent, and that I got from this Economic Times, the newspaper. Now, what is the idea of this energy independence? Are we aiming for just in one fine morning waking up and decide that we're gonna stop using coal and petroleum? No, that is not the sustainable model. So the idea is that with time, you kind of blend this the conventional energy with some of those alternative resources, including this kind of a biofuel that we are trying to generate. And that is kind of a predicted model that has been projected, like this. This brown one is 5% blending target, this one is 10%, and this blue one is 20% blending target. And as you follow this graph, this has been a projected model from 2010 to 2030, and you see that with time, the percentage of blending is going to be increased. That means with time, we'll be relying more on this kind of alternative energy, and we'll be relying less on this kind of petroleum or coal generated energy. Now, if we want to make the pledge of our Prime Minister true, then we have to find for alternative energy. And for a big country like India or China, with a lot of energy requirement in the coming years with industrial boom, it is not possible that one kind of alternative energy resource like algae or LCB or hydropower is going to be sufficient to fill this gap. So that is why, LCB can be a viable model in addition to the existing model that already exists in the market. And this is not that we are proposing that LCB is only going to be dominate the market in future. So out of LCB, we propose that bamboo has the enormous potential to be used as a raw material to produce LCB-based biofuel. And why do we believe so? There are many reasons, but a few of them to chart here is that this is a non-edible plant, so it is not connected to the food cost. It has a very high growth rate and biomass accumulation within a very short time. It does not need a lot of agronomic input like fertilizer or this kind of a thing, herbicide. So the cost of growing this plant is very negligible. It does not need like agricultural land. It can be even grown in the degraded soil. And we have a huge bioresource throughout India and many other countries which can be utilized to identify genotypes which are fast growing. Now this study, we have actually used 7 bamboo species. These are Bamboo subalqua, Bamboo striata, Tulda, Gigantia strictus, and Nutans. And these are commonly growing bamboo in India. And we wanted to explore their potential for bioenergy. So for the students, I want to tell you that today's biology, as you know, is heavily dependent on interconnectivity with other streams like chemistry, physics, statistics, biology, and computer science. And here also, since this is related to LCB biochemistry, we heavily collaborated with our colleagues who are expert on cellulose biochemistry, bioinformatics, and also the statistics to help us to analyze some of the data. So for instance, this data that I show, this was done in collaboration with Professor Jhuma Ganguly at Department of Chemistry at IIST Shibpur. Here we simply did FTIR analysis, and we want to initially have a gross understanding from all these different functional groups that what are the major biochemical groups that are mostly present in all these 7 LCBs obtained from 7 bamboo species. Now after obtaining that, from that FTIR analysis, we again went back to typical botanical analysis. So what we did a very simple kind of experimental design where we collected, so this is like a main branch of a bamboo plant, if you remember, and these are the branches. So we took the branches from all these 7 bamboo species and we simply did for a cross section, and we stained those cross sections with 2 stains. One was fast grain, which was specific for cellulose, while another one was fluoroglycinol, which was specific for lignin. And then we went for 4 anatomical parameters. One was total vascular bundle number, like in a given microscopic field, how many total vascular bundle numbers are present. And our hypothesis was that higher the vascular bundle number, higher the amount of LCB deposition will be. So that means they are positively correlated. In addition, we also measured the metaxylem lumen diameter. Now, if you look at the anatomy of this section, you will see that there is phloem, and then there is metaxylem, and then there is protoxylem, and then there is a phloem, the fiber cap, and then the LCB materials are deposited around it, right? So that is why, metaxylem lumen diameter is very important, because higher the deposition, the lower the metaxylem lumen diameter would be. Or in other words, the relationship between amount of LCB deposition and the metaxylem lumen diameter would be inversely proportional. In addition, we also use 2 other parameters, which is like a stain intensity, one for lignin specificity, another one is the cellular specificity. And we had analyzed the pixel intensity by imaging analysis, and from there we could actually come up to an integrated density value to predict where the intensity of cellulose fiber staining is higher, in which plant species or which sample, or in where the cellulose intensity is lower. And similarly, we also did the similar analysis for lignin. So total, our this experiment was done on 378 observations. And then what we did is that in addition to this kind of anatomical parameter, we also typically went for biochemical estimation. And by using this anthron-based alpha cellulose method, we measured the alpha cellulose content in all these the 7 species, and for all these species we had taken tissues from 3 different regions of the internode, that is the apex of the internode, that is the apical part of the internode, then the middle part of the internode, and then on the basal part of the internode. And as you see in this graph, that in general the cellulose and lignin content, the biochemical content, was positively correlated. In addition, when we analyze the level of cellulose and lignin in different physiological stage like apex, middle, and base, as you see here, the amount of cellulose drastically went up with the progression of the physiological age, while the amount of lignin, the progression was not so fast. Then we also had done the thermogravimetric analysis, or TGA. This was done with another chemistry colleague in the Indian ISR Kolkata, and this thermogravimetric analysis is nothing but what we did, we simply put the LCB tissue or material into high temperature up to 650 to 70 degrees centigrade, and we see that what is the temperature when the LCB composition is getting degraded. So meaning that lower the temperature it needs to degrade the LCB biomass, better it is for the bioenergy sector, because you need a lower amount of energy to get energy out of it. So as you see here, Bambusa bambus is one of the 7 species which emerge having the lowest amount of this degradation, this maximum degradation, this TG analysis. And when we compiled the anatomical parameters along with biochemical parameters to predict what is the species where the cellulose content is high but lignin content is low, because for the bioenergy sector we want a plant where cellulose content is high but lignin content is low, because lower the lignin content it is easier for the pre-treatment analysis, and it is easier that you need to spend less energy and less chemical to get the cellulose out of it. So after analyzing, we found Bambusa bambus is again another species where in terms of cellulose content it is the second best, because topmost was Bambusa nutans, but the problem is here lignin content was also one of the highest. But instead of taking Bambusa nutans, we considered Bambusa bambus as one of the model bamboo plant for future bioenergy sector, because here the cellulose content is one of the highest, the lignin content is relatively low, and the TG analysis shows that the degradation temperature is also one of the lowest. So, this study shows that combining typical anatomical analysis, biochemical analysis, chemical analysis, analytical analysis, and integrating them by high throughput statistical analysis can give you clue to the original scientific question that you had posed initially. In our case, we wanted to know that what is the bamboo species which is ideal for future bioenergy sector. Now, once we have identified elite bamboo species and genotypes, then if we want to think about the bioenergy sector, we want to develop a method, a protocol to continuously propagate that superior genotype or species. And of course, tissue culture is the immediate method to do that. And we already have standardized for these species many years ago the in vitro propagation for both Bambusa balqua and Bambusa tulda. And once we have standardized the in vitro propagation method, then we could successfully transfer them to the field, which we called field acclimatization. So the idea is, now if we want to think about a large scale production, we need to propagate this elite genotype, which has been propagated by in vitro tissue culture method in a large field area, and then try to standardize, downstream method like pretreatment or saccharification to ultimately convert this cellulose into bioenergy. Now the question is, is it really possible, or here we are kind of talking about a fiction story? But no, the answer is yes, because as you see, already a lot of activities is going on here in India, particularly in some region of Assam of India, which is already blessed with a lot of bamboo biodiversity. This Numaligarh refinery, it already inks an agreement with a Finnish firm to build the biorefinery, and successfully it could actually manufacture ethanol out of bamboo. And this is the report was published in February this year. So a lot of development has been happening in the area. So our idea is to use some of our superior genotype into put this kind of a pipeline and to develop this kind of bamboo-based bioenergy for future. So we are thinking about forming an entrepreneurship and company in future. Now, as I know that in today's lot of students are attending this seminar, I'll be telling you that one of the major thrust of Department of Biotechnology of India at the moment is empowering youth for undertaking value-added innovative translational research. Because for many decades, we have done many interesting research, we have published many high impact paper, but the question is we have to ask ourselves that what is the utility of this kind of a fundamental knowledge that so far we have generated, the scientists in the lab. And that is why I started my talk today with this notion that this is high time for us to move from model to this kind of orphan genome, because this is where the application lies. So these are the 5 youngsters from my university at Presidency University, undergraduate student. They have successfully got this funding from BIRAC funding from DBT, and they are aiming to produce bioenergy from some of those lignocellulose biomasses, including bamboo. And you are hoping that we can make some important lead in this area in future, and share with you in future about this lead, that what is the future, what we have achieved in future, and whether we could successfully convert the fundamental knowledge that we have generated in the lab into bio-entrepreneurship. So, but thing is, there are a lot of hurdles to overcome. Of course, science sometimes hear like a fantasy story, but in reality it is not. And these are the many hurdles that we need to standardize, like standardization of efficient pretreatment method. There are different factors like developmental stage, climate, and how are they affecting the lignocellulose content of the bamboo plant. We have to find a big land to maintain such large bamboo plantation, and then we have to calculate the cost effectiveness of the technology, because at the day's end, if your technology is very expensive, nobody is going to buy that technology. And for that also we have to think about the environment, the sustainability, the green energy, and that is why you need to think about the energy and toxicity audit. And these are the kind of things that we are pursuing at the moment. Now, another area that I want to share with you today is development of molecular markers in bamboo. That is another very important aspect, because as you know that bamboo is a clonally propagated plant, and when you go to the field, you see that there are hundreds and thousands of bamboo plants growing in the field, and quite often they look pretty similar. Even quite often we see that 2 or 3 different species, they look quite different, and even for a very trained taxonomist, it becomes very difficult to identify them, which species is that. And that I'm talking about the bamboo plant which are matured plant, and at the seedling stage this is almost impossible to identify. So the question that we posed many years ago is that, is it possible to use DNA based marker technology to develop a molecular marker that can be used to identify a certain species or genotype of certain commercial utility, like this bamboo's genotype I told you, which has a high cellulose and lower lignin content. Now, molecular marker, as you know, are of 2 types. These are basically DNA stretches, but this can be of 2 types. One is linked marker, where they are always linked to your target gene of interest. But in case of a DNA marker, they are actually part of the gene itself. Okay, so that is why you can develop molecular marker in 2 different ways, either linked marker or direct marker. And in order to do that, there are different kind of molecular method which are available in the field now. Some of them are PCR based, some are non-PCR based and like. Now, many years ago, the question that you asked is that, can a very simple PCR based molecular marker help to identify quality planting material, or QPM? And what do I say by QPM? Like, if you go to a nursery, you will see hundreds of such seedling, and let's see, this one is the genotype where the cellulose content is high and lignin content is low, like the genotype what we have identified. but from the morphological analysis, it is almost impossible to identify the elite genotype, right? So that is why the only way is to go for molecular marker, so that you can just go for a very simple PCR based technology to identify whether your marker is present in that plant or not, by simply extracting DNA from the leaf of that plant. And that is how quickly you kind of identify which one is a superior genotype. That we call as identification of quality planting material, or QPM. So this has been done by very simple PCR method. This is for the students. If you recall, PCR is one of the methods that we frequently use in the lab, and it has a two-step like denaturation step. The double-stranded DNA gets denatured, becomes single-stranded. Then the primers, which are specific for certain marker or gene, that goes and binds to the complementary region, and then finally comes the extension phase where the chain elongation happens. So this is how we had used a very simple method, and this work I did as part of my PhD work under supervision of Professor Amita Pal at Bose Institute, where we asked the simple question that RAPD is one of the simplest and easiest DNA-based marker that can be done in the lab. Right? It's a very simple 10 base or 20 BP long primer. So we had used a number of RAPD primers. We did PCR from all these 16 bamboo species and wanted to identify if a species specific RAPK marker can be generated for these two species, that is Bambusa balcoa and Bambusa tulda. And why did we have aimed for these two species? Because these two species are highly desirable for the paper and fiber industry, and as you know that bamboo has an enormous utility for paper industry. But the people in the paper industry, they came to us and they told us that in the greenhouse condition it is almost impossible for them to identify which one is Bambusa balcoa and which one is Bambusa tulda. So indeed we could identify like these two markers, the 836 BP long marker for Bambusa balcoa, 609 BP for Bambusa tulda, and we did southern hybridization to confirm that they are indeed species specific. And then we went at the population level study, and we could confirm that this kind of a marker can be established at the population level in different plant of the same species. But one of the major drawback of RAPD marker is that since here the primer length is lower, it has a lot of unspecific amplification. So that is why we want, in order to increase the specificity of the binding, what we did is that after amplifying this marker, we simply extended the length of the marker or primer, and we converted the 10 BP RAPD primer to 20 BP scar primer, which is now much more specific, having much less non-specific amplification. And then he proposed that these are the two scar markers which can be reliably used for species-specific identification of bamboo. Here we have done it for Bambusa balcoa and Bambusa tulda, but similar approach can be adopted to identify such marker for other bamboo species or for other bamboo genotype having different type of commercial importance. Now, from this kind of applied science, now I want to switch to the fundamental science, and I tell you that our journey in bamboo has been quite rewarding in terms of quenching our thirst to understand the fundamental aspect of the growth of bamboo and to understand the genetics of flowering of bamboo. Now, we particularly focused on reproductive biology of bamboo, particularly flower, pollen, and breeding. And why we think that bamboo flowering is so fascinating? There are many reasons, many ecological reasons, many physiological reasons, many biochemical reasons. But just to present them in a nutshell, bamboo flowering is unique. It is unique in many different respects. One is that two different type of flowering happens in bamboo. Quite often when we go to the forest, we see that large area in the forest for a big number of bamboo population, it has been induced for flowering, which we call gregarious flowering. But in other cases, for fragmented population, like the kind of population that you see around our neighborhood, there we see sporadic flowering. That is, meaning out of hundreds of plants, for some plants the flowering has happened, but others the flowering did not. So that is why, all these the bamboo flowering can happen at different extent. either at the gregarious level or at the sporadic level. And this is an ecological observation for many decades, but nobody knows that why such ecological variation exists at the population level. Another reason we think that bamboo flowering is very special is because it induces different type of inflorescences. I'll show you like 5 or 6 year old field data which we recently published. So when you go to the field, quite often we see that a typical spikelet structure which you typically see for other gramineous plant like rice or wheat like that. But at the end of the flowering cycle, we often see that a cluster of spikelet, they come together, which we call a pseudospikelets. So in the same flowering cycle, I'll show you some data. In the initial part of the flowering cycle, spikelet flowering happens, and then at the end, it switches to pseudospikelets. So again, that's an ecological mystery, that why the same plant within the same flowering cycle is switching to two different type of inflorescence habit. Excuse me. So another reason we think that bamboo flowering is very special is that here the pollen germination rate is very low. As you see here, here a kind of a 20 to 25% level by two different type of assay, we could find the level of pollen germination. And that is kind of very strange for us, because ecologically, you would assume that the pollen germination rate should be higher here, because here the flowering happens after many years, like 40 or 60 years. So we assume that when the flowering happens, it tries to accommodate all different reproductive armories in order to maximize the production of seed production, and in order to maximize its fitness, the reproductive fitness. So that is another reason why we feel that bamboo flowering is quite interesting, and there are many unanswered questions that we need to address in future. But for us, being a geneticist, we are more focused towards understanding the genetics of bamboo flowering. So we typically started with this kind of the anatomical observation, and we wanted to first understand the development of inflorescence meristem, and then the differentiation of floral meristem. And this is a typical cartoon. You see, this is a vegetative bud, where this is the shoot apical meristem, and then when it is converted to flowering or induction of inflorescence, then inflorescence meristem is induced, and then this is the middle inflorescence bud where the organ differentiation happens, and finally you get the flower with a matured differentiated organ. So that's like a typical bamboo flower. But the question is genetics of bamboo flowering. What are the genes? How are they regulated? What are the major flowering pathways? What are the genes, transcripts, microRNAs, metabolites? Are there any novel genes or that regulators that make bamboo flowering so special? Are there any special triggers like drought or physiological maturity that suddenly induces flowering in bamboo? So these are kind of a larger question that we are asking. Now today, it is not possible for me to cover all these different aspects. So I'll be focusing on 2 or 3 interesting stories that we recently published, and I feel that you may also find it interesting. So just as a recall, if you think about the physiology of flowering or how flowering is induced, like I told you, when it is timing for the plant to flower, the vegetative shoot apical meristem is converted into inflorescence meristem. That we call induction of flowering. And when does plant decide that it is time to induce flowering? That happens as a result of crosstalk with the environment, particularly the intensity of light, duration of light, cold, and some internal biochemical factor like gibberellic acid. And once plant develops this inflorescence meristem, then it is a matter of forming a mature flower. And a mature flower is developed of 4 organs: sepal, petal, stamen, and carpel. So that is the second part of the genetics of flower development that we call differentiation of flowering, where typical all these ABC genes, as you recall in your college genetic studies, that is A for sepal, A for B plus petal, B plus C for stamen, and C for carpel. So these are the ABC genes that helps in the development of organ differentiation, or more importantly, development of sepal, petal, stamen, and carpel. So again, to understand the genetics of bamboo flowering, we dissected our study in two parts. The first part, I'll be talking about the induction of flowering. What are the genes which are regulating the induction time of flowering in bamboo? And then lately, we have been focusing in collaboration with our German collaborators on some of those ABC family genes, that what are those genes and what are their evolutionary fate in bamboo. Because we believe that since bamboo is a highly polyploid genome, we can identify a lot of interesting evolutionary trend out of many of those duplicated genes that are present in this highly polyploid bamboo genome. So now in terms of induction, I told you primarily 4 or 3 pathways exist. The photoperiodic pathway, which is regulated by duration of light. Then vernalization pathway, this primarily exists for the temperate plant. Then there is autonomous pathway. There are some internal genes which regulate the flowering time. And then there is hormonal pathway, that is gibberellic acid pathway. So we primarily are focusing on photoperiodic pathway and vernalization pathway, but for today's discussion, I'll be telling you data from the photoperiodic pathway. Now this photoperiodic pathway is interesting because depending on the photoperiod, you know, there are long day plant and short day plant, they exist, and that depends on the duration of the light that the plant requires to induce flowering. And in order to do that, there are circadian regulating gene or circadian clock that exist, which are LHY, TOC1, GI, and ZTL. And in bamboo, actually, for the first time, we have identified this clock that regulates the circadian rhythm. Now, there is another gene which is known as constans, and I would request you to note this constans gene, because in our next story, I'll be telling you a story about constans and FT. So this constans is the integrator which integrates the signal that is coming from all the this circadian response pathway. Now, once constans is activated, it goes and binds to FT, and that FT is another integrator, but that is not the clock integrator, that is the pathway integrator. That is, FT integrates signal coming from all these different pathways like photoperiod, vernalization, autonomous, and GA like that. And once FT is activated, it interacts with FD and 1433, and after, as a result, this is called the Freudian activation complex and FAC, and this FAC goes and binds to the ACGT region of AP1, and then the flowering is induced. So now I'll be talking about CO and FT, and just request you to remember that CO is the clock integrator gene, while FT is the pathway integrator gene. So that we call the CO-FT regulon story in bamboo, and this CO, I told you, it goes and binds to FT, but it's a transcription factor, and its B box region actually goes and interacts with the CCA80 region present in the promoter of the FT gene. So that is why, in order to activate the FT, the B box of CO protein has to interact with the CCA80 element of the FT promoter. So then how does it happen? So first, what we did when we started doing this, no sequence information nothing was available for us, because these are the Indian bamboo plant that we are talking about, for which no genome sequence has so far been available. So all the data that I'll be showing you today, I'll just tell you that all these genes that we have sequenced by ourselves by applying different kind of targeted gene amplification method, we obtained the gene sequences, and then we designed all these experiments. So by designing gene specific primer, degenerate primer, positional cloning, all these kind of approaches, we actually sequenced 2 constans genes from bamboo satulda. One is called BTCOA and another one is called BTCOB. And when we aligned this BTCOA and COB with other known constans genes, one interesting thing we already found is that in case of BTCOB, this part of B box 1 and B box gene is absent in BTCOB. And as if you recall, the COB is the region which is important for FT to interact with the CC80 region of FT. So that immediately gives us the clue that possibly for BTCOB, this interaction ability with FT is lost, because part of this important B box 1 and 2 region are lost in case of BTCOB. But we are more interested about the expression of those genes, because this kind of a developmental study, actually expression analysis always gives a lot of clue. Now for the students, how can you study the gene expression of a gene? When we say gene expression, we are talking about the expression of mRNA, and we are talking about whether at a particular tissue stage, at a particular time, that mRNA or that transcript is being produced or not, and that we are doing by real-time PCR analysis. Okay, it can be also done by protein expression, but we are particularly focusing at the transcript level. So and usually this is being done by cybergreen. Whenever there is a double strand formation, the cybergreen grows and chelates. That means the amplification has happened. Now going back to the COA and CO story, so what we did is that we collected different sort of tissues. Some are associated with flowering, some are vegetative in nature, and we wanted to see that if there is any difference in expression between COA and COB in terms of their expression. And here two tissues are very important. One we call YLF and another one is YLM. So YLF means young leaf from flowering come. So meaning that in this plant flowering has been induced, but we collected the leaf from a non-flowering branch of a flowering come, meaning that in this plant the flower induction has happened, but that means the induction signal is present possibly in this branch, but flowering has not happened yet. While in case of YLN, this is a non-flowering branch of a non-flowering plant. In addition, we collected other vegetative tissues like come sheath, internode, and also floral tissues like early floral, mid, late, and then flag leaf like that. And interestingly, one immediate observation we found is that COA was primarily expressed in YLF, which has the floral induction signal, while COB is primarily expressed in come sheath, which is primarily vegetative in nature. So that kind of gives an initial clue that BTCO is associated with flowering, but BTCOB is possibly not. Then we were doing some literature survey, and we found that in quite many cases for other plants, this kind of a circadian response of the gene gave a very important clue. And here what we did, I want to tell you that these are all field grown plants. These are not grown in the lab. So what students did is that they took the liquid nitrogen can, they went to the field, and then in the winter and in summer, that is short day and long day, they harvested tissues at 4 different time points, the YLF and YLN tissue, like 8 a.m. in the morning, 12 p.m. in the afternoon, 4 p.m. in the afternoon, 8 p.m. in the evening, then brought those leaf back to the lab, put them in -80, extracted extraction, total RNA extraction, produced CDN library, and then went for simple real-time PCR to measure the expression of the transcripts of COA and COB. And again, we see that the expression of COA is very high at 4 p.m., which is not the case for COB. And it was already known for other published other plant system that in order for flower-specific function, constants has to be regulated in a circadian rhythm manner, and its expression has to be high in the late afternoon around 4 p.m. So that again gives us second clue that BTCOA is actually specific for flowering, while BTCOB is possibly not. But I told you that the other side of the coin is FT, because in order to induce flower, CO has to interact with FT. So that is why we also wanted to understand what is happening in terms of FT. And again, by designing gene-specific primer, we actually cloned and amplified 4 flowering locus T or FT sequences from Bambusa tulda, and we call them FT1 and FT1, 2, and 3, 4, and they are highly sequentially similar. Particularly FT1, 2 were almost 99% identical, while 3 and 4 were 99% identical. That is why when we had gone for this kind of phylogenetic analysis, you see that we got total 2 clear-cut cluster. In one cluster, FT1, 2 was aligned together, while in another cluster, FT3 and 4 stuck together. So like constants also, for FT, we also wanted to measure the tissue specific as well as the circadian diurnal response. Here I show you the data for the circadian response for FT, and as you see, again very interesting, like COA, where we got high expression at 4 p.m. Also for FT34, we got high expression at during the time point 4 p.m. while for FT12, we did not. So that again actually gives us the clue that BTCOAA and BTFT34 is actually the active COFT co-regulon, or meaning BTCOA is possibly interacting with FT3 and 4 to induce flowering, while what is the function of FT12 and BTCOB is not known yet, and we are chasing some experiment to understand what is their functional fate in bamboo genome. Now another gene that I want to discuss with you today are like CO and FT. This is another group of gene. This is known as FD. This comes much later FT. I already told you that in order to induce flowering, the CO has to interact with FT, and once FT is active, that FT interacts with FD, and once that FD is active, the BZIP domain of FD, it goes and interacts with ACGT region present in the promoter of the AP1 region. So in other words, in order to induce flowering, the BZIP domain of FD has to interact with ACGT region of the AP1 promoter. And this is how the interaction takes place. Actually, FT and FD does not directly interact with each other, but this interaction is mediated via 1433. So this is how this fluoridation activation complex looks like, and this is how FT, FD, and 1433, they kind of interact with each other to induce flowering. So again, like FT and CO, for FD also, we did the same thing. By gene specific amplification, we amplified these two genes, and we got two FD gene copy in Bambusa tulda. One we call BTFD1 and the other one we call BTFD2. And when we went for this kind of a phylogenetic clustering, we see the typical clustering pattern, what we had expected in comparison to other Graminaceous or Poaceae member. But we are more interested about the function of these two genes. And as I told you that BZIP is one of the region or domain that is one of the most critical, performs one of the most critical function to confer the FD function, because BZIP is the domain which helps FD to interact with the ACGT region of AP1. That is why, although we have scanned the other known domain like motif 1, LSL, SAP, motif 2, motif 3, SAP, like that, and we could identify some amino acid changes, but we were particularly focusing on BZIP because of its evolutionarily conserved this interacting function with AP1, and we wanted to check critically that if FD1 and FD2 has any difference in terms of this BZIP region. And this work we did in collaboration with one of our colleagues, Dr. Devrani Mitra, who is a protein crystallographer. And interestingly, what we found that indeed one amino acid, that is alanine 146 in BTFD1, was converted to leucine 100 in BTFD2. And why we are interested about this amino acid? Because when Devrani did the modeling study, she could tell us that this amino acid could be a very interesting candidate, because due to this amino acid change, the binding affinity between BTFD1 and ACGT motif may be altered by 10 times. So that is why we could predict that because of this alteration, BTFD2 may bind to ACGT motif with less affinity than BTFD1. But this is, after all, a bioinformatic prediction, right? So that is why you need to do some kind of a wet lab experiment to prove that whatever you are getting from the bioinformatic assay can be validated by wet lab experiment. So in order to prove this hypothesis true or wrong, we went for typical gel shift retardation assay, and this assay is simple. Here you use free DNA and also the protein, that is here the BZIP region, and as free DNA we use the ACGT region. And the idea is, if the BZIP domain binds with the ACGT region, then its movement in the gel will be retarded. or in other words, if the acidity is free, or in other words, if the BZIP does not bind with acidity, it will run longer length in the gel. But if the acidity binds to BG protein, then there will be a retardation or shift, or it will move slower. So that is how you can check whether this BZIP is binding to acidity or not. And in this case, we have two BZIP domain. One we got from FD1, another one we got from FD2, and we have one same acidity in both the cases. And our hypothesis is that from the bioinformatic analysis, that FD2 will bind with lesser frequency with BZIP than with acidity than FD1. And indeed, when we had done this gel shift retardation assay, this prediction was validated. We could see that BT-FD1 could bind to acidity with 10 times a higher affinity than BT-FD2. Then we also wanted to know more about the importance of BT-FD1 in inducing flowering. So that is why, again, we went back for this kind of a gene expression analysis. And for this sporadic flowering population from where we are getting the sample, we always knew that flowering was induced in spring. So that means flowering induction signal will be present in the winter. So that is why we had collected tissue YLF and YLN from spring, summer, monsoon, autumn, winter. And as we predicted for BT-FD1, we could see a high expression in winter, which we did not see for BT-FD2. That again gives us the clue that BT-FD1 is related to flowering, while BT-FD2 is possibly not. Then by the time I had a collaboration with Dr. Tony Schaffner at Munich. He is my postdoctoral supervisor, and I got a funding to went back to Germany for several months. So we decided to go for transgenic approach to overexpress these two bamboo genes, FD1 and FD2, in Arabidopsis wild type background and see what is the phenotype that we would see. And I tell you, particularly the younger students, that science, the life in science always brings you surprise. Because so far, the data that we got, we were anticipating that for BT-FD1, it is the flower inductive, while BT-FD2 is not. So that is why we are expecting that for the BT-FD1 overexpressed line, the flowering time will be accelerated, while BT-FD2, this kind of phenotype will not be obtained. But interestingly, when you went for overexpression, instead of flowering, we actually got some kind of a strange morphological phenotype. So when we had overexpressed BT-FD1, you see the growth of the leaf was very much reduced, but in case of BT-FD2, it almost looked like a normal plant. So we scratched our head for many months that what is going on and all these things. And then we wanted to understand that in terms of kinetics, or days after germination, that when is this difference happening. And we took, actually Sritikona took this tedious, this epidermal peel assay, measuring the size of the epidermal peel of wild type, the overexpressed BT-FD1 and overexpressed BT-FD2 plant. And then we see that this difference in size is primarily due to different in cell expansion or growth, and that primarily happened in a timeline between 22 to 30 day post germination. And we explained it in a way that in case of BT-FD1, indeed the flowering time was little bit accelerated, but actually this vegetative growth, we possibly this suppression of vegetative growth we possibly get due to too much overproduction of FD1. And it is already known that Arabidopsis already, wild type Arabidopsis already had a native copy of BT-FD1 gene, but it did not have a BT-FD2 copy. Okay, only rice has 2 BT-FD copy, and also in bamboo now we see 2 BT-FD copy. So the suppression of vegetative growth is possibly a secondary toxic effect of overproduction of the FD protein. But from the transgenic analysis, we could get the clue again that indeed FD1, the flowering time could be accelerated to some extent. So again, it proves that BT-FD1 is involved in flowering while BT-FD2 is not. The last part of this flower genetics I told you is related to floral organ differentiation, like this development of all the sepal, petal, stamen, and carpel. And this is the typical model, ABCD model, by which we can explain. And to understand this genetics, we are chasing some of the Matchbox genes in collaboration with Professor Annette Baker at Munich, at University of Gießen, Germany. So what Midustri is doing, she is chasing all these huge number of Matchbox genes in different sequence bamboo genome and trying to understand the evolutionary origin, the mode of duplication, the rate of synonymous versus non-synonymous range, synteny, all these different kind of evolutionary analysis to understand the evolution of Matchbox gene in bamboo, which I'm not sharing today because this paper has not been published today, and I hope that very soon I'll come up with that story with you and I can share with that today. But a very small part of the small story that Sukarna did in her PhD thesis is that I can show, this is also under preparation now, is that this MATS14 versus MATS15 story. These are two MATS gene, and again in bamboo we see that in terms of floral induction versus floral organ differentiation, again we see a different kind of a trend which we do not usually see for other typical crop or sequence genome. Right. So that again I want to emphasize, like the way I started my talk today, is that when we started working with bamboo, we know that our path is going to be rocky, but we always wanted to give a try. And so far our understanding is that we feel lucky that we decided to take this rocky path because it actually gave us the opportunity to identify many interesting genes, their duplicated copies, their new regulation, their new phenotype, and we hope that in future we can come up with many more genes and their altered regulation. I want to end my talk today little bit talking about breeding in bamboos, because my title of my topic is genetics, genomics, and breeding. As you understand that this is a vast topic and this is almost impossible for one speaker to cover all the aspect. But since I've been working on bamboo for so many years, so I thought maybe I can enlighten the audience about a little bit progress that we could make in the area of bamboo breeding. Again, with humility and with a lot of humble approach, I say a little bit progress we could make because we did not contribute a lot in this area, but we are trying to understand some of the genetic pathways again related to genetic compatibility in bamboo. Now, when we talk about bamboo and when we talk about breeding, people get really scared. And that cannot be easily anticipated. That can be easily anticipated, right? Because this is a tree plant. This is not like a small plant that you can grow in the lab. Here the flowering is infrequent, which may happen after 40 to 100 years in a range. It is sporadic. The pollen viability is very low. The germination is also very low. And in our study, we found even that this in vivo germination, the timing of the day is very important. So it varies which time of the day you are doing the in vivo germination. The plant is clonally propagated, so that means it is very difficult to select the two parental genotype that you would like to cross, right? Because you do not have any genetic information about those plants. You do not have any idea about their physiological age, their level of genetic heterogeneity, because many of them are cross-fertilized. So that is why it is very difficult. It is very challenging to aim for bamboo breeding, but still we aimed so, and I can at least show some preliminary data, and we find that these are interesting, which we recently published in a journal. Now, as I told you already, that this is the data that I'll be showing you that we did for last like 5 or 6 years. It's a kind of field-based breeding experiment, and I'm really thankful to my students, my hard-working students, that they agreed to do this hard work, because nowadays usually students are mostly focused on the lab-based genetic work. But when I came up with this proposal, they accepted this proposal with a smiling face. So this study was done in a range of 2015 to 2019, and then as you understand, this arrival of COVID and all these things, thing got really complicated. So we did actually studied this population. One was in Shamnagar 7, Shamnagar 16, and Bandel 23. And in the entire flowering cycle, we assessed the number of true spikelet versus pseudospikelets. And one interesting observation that we found is that, like I told you before, that during the end of the flowering cycle, it was always the pseudospikelets abundance that was higher than the spikelet abundance. And in this paper recently published in Plants, we come up with some theoretical ecological explanation that why this might have happened. Then we also see that in case of pseudospikelet, that the amount of seed production was much higher compared to the true spikelet. But more interestingly, we also see, well, more interestingly, what we wanted to do is to go for this kind of a breeding or crossing experiment, because not much crossing experiment has been tried by people in the field, and we wanted to give a shot to see how does it go. Now, one of the challenge when you are doing for this kind of crossing experiment to ensure that the two parental genotype that you are selecting, they are indeed genetically different, because as you know, bamboo is clonally propagated. It is always possible that this clone is actually grown by obtaining some clone or vegetative cutting from this plant, right? So that means they are genetically very close, or from originating from the same donor stock. So that is why, you know, we have selected two populations which are physically far away from from one other, and then in both cases we did selfing, like we took the pollen from the one plant and did put on the stigma the pollen dusting on the same plant, and then we put a plastic bag to stop future cross pollination. So this was for selfing, and for crossing we took the pollen from one plant, and then in the physically distant population we did the pollination experiment. Now we also did some additional studies, such as starting from 6 a.m. to 2 p.m. we wanted to know what is the time point when the number of open florets are maximum, because that will maximize the possibility of this cross fertilization. And we see that 10 a.m. was actually the time when this fertilization rate was highest. And then we did both selfing and crossing, and interestingly we found that for Bambusa tulda, we primarily call it as a cross-pollinated plant, because here the rate of cross-pollination was in a range of 32 to 41%. And that, in this paper, we came up with again some kind of ecological explanation that it makes sense to us that since Bambusa tulda is primarily a clonally propagated plant, and since here the flowering cycle is very delayed, very long, so that is why, you know, it always tries to kind of possibly maximize its cross-pollination efficiency so that it can induce some kind of genetic heterogeneity in the genome or genetic diversity. Now, how is this kind of genetic, the cross-pollination is happening? Is there any physiological reason behind that? And we could actually identify one physiological reason behind that bamboo is attaining. This is we call protandry. So here, what happened when we did this progressive or developmental analysis of the flower and different floral organ, we see that androecium in the flower is always maturing before gynoecium. And as you see here, when the gynoecium is matured, actually the androecium, the stamen, this anther, they're already withering, right? So that is the physiological situation the Bambusa tulda is creating by this kind of a protandry, that differential rate of development between androecium versus gynoecium. And this is how it is forcing itself to go for this kind of a cross-pollination, a high rate of cross-pollination. And this is possibly the last slide of my slide. This is another area where Sukarna and other students in the lab, they are trying to make some progress. We now are trying to understand the genetic pathway regulating the sexual incompatibility in Bambusa tulda. And as you know that in plant, there are two types of genetic compatibility pathway exist. One is gametophytic self-incompatibility, and another one is sporophytic self-incompatibility. And we believe that in bamboo, like in other poaceae member, possibly it's the gametophytic self-incompatibility or GSI that exists. And we believe that these are the pathways, that is SLF and sRNase mediated pathway, that plays a very important role to confer this kind of a GSI mediated sexual incompatibility in bamboo. But again, this work is currently underway, and I hope soon we will be able to come up with an interesting story out of this study. Last but not the least, as you see, that this is where I got the title of my talk: Genetics, Genomics, and Breeding of Bamboos.
Excuse me, Malay.
Yes.
Slide sharing, I think there is something is happened because in YouTube, the current slide is not visible. It's stacked somehow or anything.
Oh, I'm so sorry. But those who are online, can you see it?
Yes, I'm no, actually in Zoom it's not saying. I think you should
The slide that we are able to see is ectopic expression of BTFD1 repressed vegetative growth.
It's a long long back it was in the,
So FD.
yes. So you can just start slide sharing once again.
This one, right?
Um,
This is the one probably you have seen last.
No, but we can. Here it is.
Yes, slide sharing is
sharing
somehow
has
stopped.
frozen. It is frozen now. So we have to get off and get on again.
So resume share. Okay.
Yes, resume share, or you can start sharing, stop and start again.
Yes, now it is. Yeah, it's moving.
Okay, so I think up to here it was okay, na?
Yes, no.
This is the yes. We'll just have to go from here. Yeah.
Here.
Yes.
From here. Okay. So here I showed like this overexpression data that the vegetative growth was highly stunted in case of BTFD1, while in case of BTFD2, no such phenotype was observed. And then by epidermal peeling analysis and analyzing the epidermal cells, we could see that this kind of difference in growth was achieved due to differential cell expansion, and that primarily happened in a time range of 22 to 30 days after germination. And then I was also discussing about this ABCDE, the floral organ differentiation genes in bamboo, and we are studying on this this AP1 gene family and this Matchbox gene family in collaboration with Professor Annette Baker's group. This paper is currently under preparation, but we hope that quite soon we will be able to come up with data for the first time in bamboo genome. We will be discussing about a huge number of Matchbox gene, their evolutionary history, the mode of duplication, the rate of synonymous versus non-synonymous mutation, their synteny, and all different kind of evolutionary trend that is happening in such a polyploid genome. And more shortly, here from Sukarna's thesis, I can share with you that this Mars 14 and 15, when you zoomed in, we see that in terms of expression, and when you deduce their possible function in terms of flower induction, vegetative development, and floral organ differentiation, they are quite different compared to its evolutionary related genome like rice, brachypodium, or even Phylostachys, the other BAM. Then this is one of the last part of my talk, like this breeding aspect of bamboo I was discussing. And I told you that not much people aim to study the breeding of bamboo because of many reasons. Obstacle such as its tree habit, infrequent and sporadic flowering, the very low rate of pollen viability, the timing of the day that affects the pollen germinability, the clonal propagation method, which is leading to lack of information about genetic background, which is very important to select the parental genotype that needs to be used for crossing, then their physiological age, level of heterogeneity, like that. But still, we tried some effort for 4 years. This is the data I show you. The students went to the field and they studied the flowering cycle, the recurrent flowering cycle or sporadic flowering cycle, population of Bamboo satulda, 3 population. And that gives us a lot of interesting simple ecological data. Like I told you that 2 different type of flower inflorescence we could see in a single flowering cycle. And when you compare their abundance or frequency, during the end of the flowering cycle, the abundance of pseudospikelet was much higher compared to the true spikelet. And when we counted the seed number, we also see that the number of seed was much higher compared to in the true spikelet compared to the pseudospikelet compared to the true spikelet. And for this, we came up with some kind of ecological and possible evolutionary explanation, which we explained in our plants paper, which we recently published. But this is what I want to kind of end today, is that yes, it is possible to do breeding in bamboo. We are one of the few groups which tried to do this kind of crossing experiment on the plant, which are growing in the field. And we selected these two population, which are around 340 meter, growing from separated from each other, to ensure that they are not generated from same clone, but they are of different genetic origin. And then we did both selfing and crossing. And for selfing, we took the pollen and did the dusting on the stigma on the same plant, same flower. But for crossing, we took pollen from one plant and then dusting on the flower of the another plant. And immediately after pollination, we put the plastic bag to avoid cross-pollination. We also did some kind of a fundamental question like, is there any difference in terms of a number of open flow rate with time? And indeed, we see that 10 a.m. is kind of optimum time when the maximum number of flow rates are open. So that we recommend that if anybody wants to do this kind of in the field, in vivo crossing experiment, probably 10 a.m. in the morning or until 12, maybe the optimum time to do that. And our original question was, is bamboo setulda is primarily a self-pollinated or cross-pollinated plant? And our data indicates that this is primarily a cross-pollinated plant. And we explained in a way that since bamboo setulda is primarily clonally propagated, so possibly it forcing itself to cross-pollinate to increase its genetic variability. And one possible mechanism or physiological mechanism by which it is forcing itself for cross-pollination is this kind of protandry, where the differential rate of growth between androecium and gynoecium has been observed. As you see here in this slide, we did in the developmental progression of flower, and we see that the rate of growth of gynoecium, so androecium is maturing much before than the gynoecium. So when the anthers are kind of dry, then the gynoecium is ready for pollination, right? So that is why physiologically also it is not possible for the plant to go for self pollination. But nowadays we are more interested to dissect the genetic pathway or the genes that is controlling this kind of genetic incompatibility in bamboo. And in plant, primarily two genetic incompatibility pathway exist. One is called gametophytic self-incompatibility, GSI, and another one is SSI. We believe that in bamboo, like in other poaceae, the GSI pathway exists, and out of many pathways, we already got some clue to believe that here this SLF versus sRNA's kind of interaction is playing a very important role to control this kind of a GSI. And I hope that within soon we should be able to come up with some interesting story out of these genes and data that we are getting in the lab. So this is where I was just telling that this is the title of my topic, Genetics, Genomics and Breeding of Bamboos. And this is actually a title of a book that we recently published. It will be open in the market in April, sometimes around March or April next year by CRC Press. And those who are particularly interested about bamboo biology, I'll request you to go for this book. Here are many groups from China and South America and Africa. They have contributed and enriched us to understand a different aspect of genetics, genomics, and breeding of bamboos. So with that, I would like to stop today, but it would be an injustice if I don't thank my hardworking students who are actually generating data in the lab, and I am just presenting the data on their behalf. So we have a very dynamic group composed of course, PhD students, number of PhD students, but also masters and undergraduate students. So Shubhadeep and Tauhidur, they are primarily involved in the lignocellulose research in bamboo. Midushree is working on the vernalization pathway and Metz box genes. Sritikona has already got her PhD degree. She immensely contributed in the constants and FD study on bamboo. Shukunna did a lot of hard work in this crossing and Metz 14 and 15 studies. And then Proshun, he did studies on FT and pollen viability study. And there is, I don't see Shonali, she is now working on the microRNA mediated regulation of bamboo flowering, the data which I did not show today. Professor Amita Pal, I'm immensely indebted for introducing me to such a fascinating plant. I did her PhD under her supervision on bamboo, and then after a long postdoc career, I went back to India and again continued my studies on bamboo. Dr. Devrani Mitra, my collaborator on protein crystallography, Dr. Tony Schaffner, and Professor Annette Baker, my German collaborators, who are helping me in many different ways with transgenic and evolutionary studies. And of course, last but not the least, my funding agency is my university, Presidency University, Kolkata, DBT, CSIR, UGC, and Alexander von Humboldt Foundation, Germany. So with that note, I thank you all for all your kind attention, and I'll be more than happy if I can answer or address your question if you have. Thank you so much.
Thank you. It's a great presentation with the depth, knowledge, and information. Many people appreciate your presentations, and as you know, that many people ask many questions, which is some beyond of your today's talk, and some are related to today's talk. So I'm collecting all those questions. I'll go after your interview session. So
Sure.
So there are a few questions related to the biotic related, the disease related on the bamboos, but today we are not include this talk, so we are not going to take all the questions. Many people are interested to the breeding and tissue culture, and some people are also start from what we have initially thought about the bamboos. One interesting is that yes, up now I know basically bamboo is the favorite food of Kung Fu Panda. But today only I got to know the real importance of it. Wow, really very insightful session and very knowledgeable. So yes, it's happened. It happened when orphan crops are a rare crops, someone working. It's very challenging and at the same time very fascinating for the other society and other scientists. And I personally know his lab, means today's Malay, Dr. Malay lab. So I know that people are very hard working there, and very interestingly, they are working in such fields, which is very difficult. Sometimes I also wonder that how they collect the flower from such a long tree, and the cycles. When you're working on the cycles and time point is very difficult, because you have to go there and time. Because here is a cycle, as I saw in your slide, there is a cycles, morning 8 to night 8 to sample collections. So
for
must
the Circadian.
Yes, analysis. So must be they going to the field around 6 p.m. and returning lab at 10 p.m. to freeze the samples. So it's a hard working people, otherwise you can't generate. It's look like a simple data. But sometimes doing this simple thing is very, very much tough in when the conditions are not related to your favorite lab, because it's most of the in field based. So I request Soma, and what I saw in Zoom, there is maybe Professor Amita Pal is there. I don't know if she is there. We will glad to ask introduce her also. Let's see. Let's first I ask Shoma to go for the interview session. As per now, I collect 4 questions for your interview, but I collected all the questions from YouTube Live.
Okay.
And they are very much interesting. We are now 240, 41, or is
Wonderful.
increasing. They are live. They are also live. So we are going for the interaction first and collecting.
I'm sorry for this disturbance with the slide change. Actually, I
No,
didn't
no, no,
realize
no.
it.
It's happened. It's
Oh, it
happened.
happens.
It's happened many times.
No worries.
No worries. No worries. Soma, please start.
Yeah. Okay, so I was watching the YouTube chat, and people are appreciating it a lot.
Yes, yes.
Successful session. So we will start with a few questions. I call this session to know the scientist better, how we can get to know the scientist better. We know the science and now we get to know the scientist. Sir, we will be very happy to hear how you paved your path to become a scientist and how you got into bamboo research.
Yeah, like I was telling, I started my career as a PhD student in Professor Amitabh Pal's lab at Bose Institute. And there first I was introduced to bamboo. And at that time, during my PhD tenure, I did the tissue culture and this molecular marker thing that I showed today. But I was fascinated with kind of the diversity and complexity of biology that bamboo can offer. And then, as you know, typically after completing PhD, I went for postdoc, for a long postdoctoral career, actually 2.5 years in Oregon, in Environmental Protection Agency, USA. Then 1.5 years, I was involved in a parasite plant genome sequencing project at Virginia Tech. And then I got this Alexander von Humboldt postdoctoral award to move to Germany. There I was working more on fundamental gene duplication and how it affects our ability to predict gene orthology, functional equivalence, all this fundamental aspect. And then after that, I came back to India with this position as assistant professor at Presidency University, Kolkata. And then I started thinking about my own group, about the kind of research. Yes. Am I audible?
Yes.
Yes. So started thinking about the research, what I'll be doing. And then I realized that I should be doing something which is relevant in the context of India. And that is why I was emphasizing on this model plant thing, because in US, mostly I see myself and majority of the research group, they were stuck to the model plant. But then after returning back to India, I realized that it doesn't help me, maybe to some extent, to understand the biology of the plant. But I should really challenge myself to move to a plant which can offer some kind of economic importance, and to see the kind of training that I got in US and Germany, to apply some of this training to some of this kind of orphan plant, to see if it can help me to identify the question that that we are trying to generate. So with a very small lab in a small university, we started. And then, like I told, that the students are very hardworking, so we keep trying. So that's kind of like background and genesis of how I started my career with bamboo. I disconnected from bamboo research, and then again I got reconnected to the bamboo biology.
Sir, what are your thoughts on natural plant-based fibers and their future in industrial and commercial applications?
Yeah. So, like I told you, there is a lot of interest lately on bamboo. I'm sure also in China. Recently, we had this bamboo the bamboo conference meeting in China. I hear from my colleagues a lot of enthusiasm because of this. The UN also declared bamboo because of many of these SDGs the bamboo can attain, and one of them is actually this fiber. Because bamboo is being used in the paper industry for a long time because of its enormous potential to contribute fiber. But then there were many reasons because of availability of the stock and so on. There are some dip, but I think with the systematic studies and with the kind of study that I showed you today, identification of this one bamboo genotype having high cellulose versus low lignin, because for fiber industry also, the requirement is similar. They want bamboo plant which has a fiber which has a high cellulose versus lower lignin content, because if in the fiber the cellulose content is high, its tensile strength is higher. So that is why, so similar approach can be used. And like I told, that we have such a huge biodiversity, genetic diversity available in India, China, and some southern part of America. And I think we did not even utilize even 1 or 2% of that genetic diversity. So with this advancement of knowledge and technique, if we can form like a global larger consortium, and if we can exchange ideas and collaborations, I think it is highly possible to explore the global genetic diversity and to identify some of those genotypes which have the potential to produce, elite this kind of a fiber yielding plant.
Right. Sir, what motivates you to work for the betterment of agriculture?
Yeah. So as you see that I'm not directly an agricultural biologist, but as a plant biologist or botanist, I have, of course, a lot of interest in agriculture. And in another aspect of my research, which I did not show today, we are also working on Brassica, which is the mustard family. And in India, one of the major problem of Brassica research is salinity, because a big part of the agricultural land is getting saline because of different type of natural calamity. So the salinity is a problem. So here what we are doing, actually, we are studying the the genome wild relative of Brassica, like Thelongiella halophylla, which are highly salt tolerant, and we are applying different kind of genomic tools to identify duplicated gene copies. In fact, for HKT gene copy, we already have found 2 such gene, which we published recently in Journal of Plant Physiology. And our idea is to use basic bioinformatic pipeline to identify such duplicated gene copies in the wild extremophiles, which are related to the crop plant, to identify those gene copies and to transfer them back to the crop plant by transgenic method. And this, we believe, can be one potential approach for future to identify novel gene, novel stress tolerant gene. And this is how we can help the agricultural scientist to confer tolerance to the crop plants, including Brassica.
Which sectors of plant science research do you think will prosper in the near future?
Yeah, so I think research in future and research funding in future, there will be less public funding and there will be more public funding. And we all have to think about this changed global reality. And we all have to try to adapt ourselves to change global realm. And that is why we have to think about the applied aspect of the plant biology. I'm not saying that the fundamental aspect is not important. We are also interested in fundamental aspect. We show you many data how the gene function can help us to identify new gene and use some of those genes for improvement of crop. But for funding, or in order to get funding, we have to show some kind of applicability, and that is why we are gradually moving towards this energy research, because for plant scientists, I'm sure that this energy research will give a lot of leverage in future in terms of finding or exciting finding. And another thing, as a scientist, we should always be able to explain to common people that what we are doing in the lab. Now, this kind of energy research, everybody understands its importance, starting from policymakers to government, to administrators, to common people in the society. So I think that is why this kind of energy research or healthcare or crop improvement, I think these are the major areas, because everybody understands the global food security. So that is why crop improvement is another big area. The stress biology, the stress area. So these are the areas, I think, where a lot of emphasis is being given. And I'm sure in future, a lot of new emphasis will be given by the funding agencies and also the policymakers.
Talking about the future, which sectors of plant science research do you think will prosper in the near future?
Yeah. So again, I think the area of plant stress biology, because crop stress is a big area, and as we understand that our population is increasing, but the agricultural land is decreasing, and also many of that agricultural land is becoming inagriculturable because of salinity and all these things. So that is why stress biology is one big area. The energy biology is another big area, and of course this the medical biology, like all these fatal diseases, particularly in last few 2 years or so, I see a lot of emphasis on viral diseases, including COVID and all these things. So I think, but recently I also think that the metabolomics, because application of computer programming to design different type of metabolites which may have ability to control the plant architecture, which may have ability as a drug molecule. So this kind of a chemi-informatics, where we combine the knowledge of chemistry, bioinformatics, and biology to come up with a product which will have a biological implication. That is also another area, and I'm sure a lot of private partners and funding agencies will be interested in these areas in future.
What are your thoughts on climate change and global warming effects on agriculture?
Yeah, so climate change is a real thing. There is no way we can shy away from it. And as you all know, this global climate change and greenhouse gas effect. So this is high time that we take really immediate step for that. And that is why we are proposing of this kind of a bioenergy. And what we feel that in a big country like India, it is not possible to propose one solution as a viable solution for the entire country. For instance, like a hydroelectric, or turbine from the wave generated from the ocean, or wind. So what we feel is that the Niti Aayog or this kind of organization may take a holistic look to identify or divide the entire country in different sectors, and depending on the availability of different kind of sources of energy, we can think about, like you see already in Punjab, every year during this crop harvesting season, farmers are harvesting the crop, and then the residual part they are burning, and that burn is creating a lot of CO2 gas emission and carbon dioxide effect in the neighboring region. So I recently heard DBT actually bought some kind of this kind of mobile machine, and they are trying to give it to the farmers so that they can cut it, and they can at a local basis they can generate a bioethanol or this kind of a scene. So that is the thing. The science has to be holistic and realistic and timely, and we have to make our policy, and we have to integrate science in a way so that this issue of real climate change can be attained, because only just changing the policy will not change the ground level situation. So that is why it is not only the policy, but it's also the technology, the cost of that technology, the public perception, the government policy. All these things has to be integrated to make it more meaningful and being more functional.
Sir, talking about policies, this thing comes to my mind. What is your opinion on GM crops, and do they have a future in attaining food security?
Yeah. So I shared with one personal note a long time ago during 2005 or 6. At that time, I started my postdoctoral career at Environmental Protection Agency, EPA. And EPA is a federal lab which actually regulates all kind of environmental issues. So at that time, two unique problems came to the agency. One was this nanoparticle, and another one was this kind of a transgenic plant. And then the federal government asked the agency to assess that, because this is the time when nanotechnology was evolving, so cosmetics and all these things, lot of nanoparticles were being added, and then some of it were being thrown in the environment. So the government and the Greenpeace movement, they were worried, like, if this kind of a nanoparticle may have an impact on environment. Similarly, For transgenic also, the government wanted to know that the release of this transgenic in nature, whether it may have any unintended effect at the ecological scale. So that is why this paper, my neighboring labs, the first time they published in PNS, the landmark paper, where they at a ecological scale, they designed this experiment where they introduced the transgenic plant, and they showed that in fact the transgene can flow in wild relative, and hybridization can happen, and that flow can happen towards the direction of wind up to few kilometers. But given having said that, I would not think that transgenic is really kind of a big worry to release in the nature, as far as we do sufficient ecological and physiological test in the environment. And when we are growing, particularly if we grow some kind of a marker-free transgenic, I don't see any reason why we should be kind of hesitant to release a transgenic plant to the nature. Because what people are arguing is that, the nature itself is a laboratory, right? So the polyploid genome that we are using nowadays, they also are product of some kind of transgenesis. Now, I know you will not call it transgenic, because this has happened over millions of years, and it has tested over the selection pressure of nature. But in principle, in terms of biology, there is not much difference. And if we really want to secure food security of the world, I think transgenic is the only way out. But again, we got to be careful. We need to do all the sufficient QCs, particularly with reference to ecological impact, to make sure that it will not have any unintended effect on the non-target organism, like other plant or bacteria or mycorrhiza or like that.
Sir, what is your opinion on digital agriculture, and is it wise to combine artificial intelligence and agriculture?
Yeah, I know Gates Foundation is funding as a lot of fund actually in Nepal, and I think in India also. Many years ago, I had some discussion with some of their strategic partner in Asia. So this is really important. So at that time, I heard some kind of a technology, like some kind of imaging IR technology. They were putting it below a drone, and they are flying those drone on top of agricultural field to assess like the level of humidity or different kind of a nutrient concentration in the soil. And this one I call is kind of more like a similar thing what they're doing in the precision medicine in medicine. So for agriculture, I think it is very important that we enable our farmers, and this is part of that enablement, right? The farmers need to know that how much will be the rainfall in next 5 days, or what is the nitrogen concentration or phosphate concentration in the soil, so that whether I need to apply more NPK or not. So this kind of local information, I think, is very important, and I'm sure this kind of information is available somewhere. But I think there is not such a holistic approach to integrate all this information and to provide it as a app or something to the farmer, so that at any given day, they wake up in the morning and they can decide that whether for next 5 days or so they need to be worried about watering in the field or not, or whether in the next 1 week or so they need to apply any more NPK or not. So that will have another impact, that will show the real power of science to the society. And I think that is the only way the scientists can convince the policymakers and the bureaucrats that science really works in our daily life, and we can pressurize them to increase the funding and their funding in education and research and all these things that all the educationist is crying for such a long time.
Sir, very well put, sir. Sir, now I'm coming to some questions for that concerns the students. Sir, how has been your experience working with students in plant sciences?
Yeah, so I actually studied botany, both BSc, MSc, and then I did my PhD on plant. All my 3 postdoctoral career, I also worked on plants. So all throughout my career, I have been a plant person, and I feel lucky and fortunate that I studied plant, because I think that's really a natural science or part of the natural science, and that can kind of answer your curiosity and your many real thing that you see around you, and that can help you to answer. So that is my message to the students who are studying plant science, that I think this decade is the decade of biology, and the next decade I'm sure will be the decade of the plant biology and agriculture, because everybody understands the importance of food, everybody understands the importance of energy, everybody understands the importance of clean nature, and we, the plant scientists, can make a big difference in all these different areas of early walks of life. So that is why it has been fascinating. So far I have got a lot of good students in our university. I get we teach we have a life science course, and part of life science course we teach plant biology, and I find a lot of enthusiasm among the younger students about different type of physiological phenomenon, particularly when you go for excursion. When you go to forest and you're taking a walk through the forest with the student, students observe many things, and they ask many different questions, and to be frank, most of the times we are unable to answer the question. But that is the beauty, right? We always learn from each other. It's a reciprocal learning process. We are also trying to learn from the students. We come back home, we study more, and we try to get back to them with some modest answer. So the experience has been fantastic so far.
Sir, what advice would you give your students when they find it hard to decide between joining research and the industry or corporate sector?
Yeah. So that is a typical question that I always hear from my students also. And the thing that I tell, when we were students and when we were preparing for our future, majority of them, I'm sure, of our generation, we are always aiming for secured government jobs. But the time has changed, the generation has changed, the government jobs has reduced, but at the same time the private job have been reduced. Now there is a big area of entrepreneurship, the company formation, the business model, and like one initiative I showed in my talk with the undergraduate student, we are also even trying with one of those pilot projects. So that is why my message for the students is that now it's a broad globe. The horizon has been widened, and I think you should keep your eyes open, and you should keep for all different type of opportunities, and not only just typical research or job. You can also think about some kind of entrepreneurship development, small ideas that can be converted into a technology that can be patented, and that can form future technology development, right? Just think about the person who had come up with this concept of Ola or Uber, right? So this company, they're not buying any car, they're just providing the platform. So I think it's the idea in 2022 or later, it's the idea which will matter. And there's not a lot of funding from government, from bank, and all these things. So just be aware, be vigilant, and explore new areas. And I'm sure that many of you will succeed in your endeavor.
Oh, that was wonderful, sir. When you hire a researcher or a fellow, what qualities do you look for in that person other than academics?
Yeah. So I always tell my students, we always stay in the lab. For the PhD students, I mean, from 10 a.m. to sometimes they stay like 8 or 8:30 at night. So sometimes we even stay more in the lab than in the family, right? So that is why lab becomes our second home, and you have to find a way to love your lab, because whatever you are doing, if you don't have a passion for it, you can never excel in whatever you are doing. So that is why the first criteria when I select a student or group member, the first criteria I put emphasis on is the integration ability of the student, because today's research you cannot do in isolation, right? You have to do it in a group, in collaboration. So that is why you have to have a kind of communication ability. You have to have a good temperament. You have to be hardworking, and most importantly, you should have the mentality to accept failure in research, because in biology research, majority of the times experiments don't work. It is not a fantasy world. So that is why when a new student join my lab, I always warn them that be prepared that majority of the time experiments will not work. But you should stick to it. You should think your analytical mind that why it didn't work, so that you next morning you come with a fresh mind and you try to solve the puzzle. And then after a few effort, when the puzzle is solved, then the kind of a fun or excitement you get out of it. That is why we are doing research. We are not doing research for money or fame or anything, but I think it's all about passion and it's all about fun.
It's amazing. Sir, so we are at the end of this session, this wonderful interview session. So as a note to end this, what would your words of advice be to the students and scholars who are new to the field of plant science research?
Yeah, so my only advice will be like I was telling, that don't get biased to any particular thing, because quite often I see students come, and when I ask them what you want to do, they say I want to do molecular biology. Molecular biology, biochemistry, DNA recombinant technology, these are nothing but a method. This is just a tool, right? But most important is you need to know
Nowadays, sir, there is CRISPR craze.
CRISPR, yeah. So this kind of a fascination, students are fascinated with this kind of a fancy word. But I showed you in my own data that I collaborate with, starting from chemist to statistician to bioinformatician to biochemist. But I don't do collaboration to do CRISPR-Cas or to do a certain crystallography, a certain technique, right? I always do the collaboration to answer a question. So that is why I tell the students that first ask the question, that what is your question, and then try to find out what is the easiest way or what is the modest way or cheapest way to solve that problem. So don't be too much fascinated by techniques, methods, or certain cool stuff like cancer or CRISPR. Just ask yourself what you want to do in life, and if you feel like really research is your cup of tea, and then you feel yourself that whether if you are fascinated by plant science or not, and then just find the right lab and ask a good scientific question, put your dedication and your all heart into it. I'm sure if not today, at least tomorrow, you will have the success.
Right. That was amazing and very insightful. I'm sure the students are absolutely agreeing and loving this session. So now I think it is time to answer a few of the questions that we have collected from YouTube. So Shubho,
Yes,
have you
highlighted
finished
one. Yes.
collecting? Okay.
Four highlighted questions.
So can I ask them?
Yes.
Okay, so Masood Majaz Jane, I think I don't know if I'm saying the name right, is asking, are all the species growing in tropical and subtropical regions?
So the species that I was talking about, yes, they are mostly growing in tropical and subtropical regions, including India. Primarily, we are focusing on Indian germplasm because that is available to us. So they are primarily, yes, this Bambusa genera, they are primarily dominant here in this tropical region.
Okay. Then Rakesh Chaudhary asks, sir, you studied the function of bamboo FT-FD genes in wild-type Arabidopsis by overexpressing them. Sir, why didn't you study in Arabidopsis FT-MD mutants, though they are available?
Yes, that's a very good question actually asked. So what happened actually for FD, for instance, this null mutant, it is very difficult to get, because if you if you go for complete loss of function, the plant will not produce flower, right? So similarly for FT, actually Arabidopsis has two copies, FT and TSF. So you can get like a single mutant, either FT or TSF. You get some phenotype, and then you can complement. So that is what we are trying at the moment. So in that paper, we actually went for overexpressing then in the wild type Arabidopsis, which already has its native copy of FT and FD. And in case of FD, like we said, because of overproduction, there was a toxicity effect. So that's a very good question, right question. We are actually going for it to complement some of those single mutant Arabidopsis lines with bamboo genes.
Okay, so Rukaya Memon asks, what is the scope of growing bamboo species in plant tissue culture and what should be considered while doing research?
Yeah, so when I was doing tissue culture, I did tissue culture on bamboo for quite some years. I did the tissue culture by clonal propagation method from axillary bud meristem. By using cytokinin and then auxin, we induce the shoot multiplication. But there are a lot of problems that we face. So when you are using mature tissue, one major problem is the endophytic fungal contamination. Because when we are doing tissue culture, we are sterilizing the surface. That is why we are calling surface sterilization. But the problem is you establish the culture, multiple shoot multiplication, you get the plant, but after a few weeks, suddenly you see the fungus is coming out from inside because there is some endophytic fungal contamination. So another problem is this the phenolics that are associated with this kind of a mature tissue. So to avoid that, we use some kind of antioxidants like PVP or PVPP that typically people use in the lab, and that gave us some good result, but that's another challenge. And another thing that also reported in that PCTOC paper is, interestingly, we found when we are collecting this nodal explant from the mature plant, we found that there are some season where when we are doing the tissue culture, the rate of in vitro multiplication was much higher compared to the tissue which were collected in other season. So we believe that possibly it was connected to some kind of endogenous hormonal level or so, which is probably influenced by the this kind of a seasonal condition like temperature, heat, light, like that. So yes, to answer your question, it is possible, but to do it from mature tissue, they are like this kind of endofungal contamination or endogenous phenolics, this kind of a problem that you may face.
Okay. So there is one other question from Gautam Chabra. What will be the perfect timing for selfing or crossing in bamboo? Means time when there is more pollen viability.
Yeah, so we did this kinetic study, and in the field, actually we found that usually in the early or late morning, right, 10 to 11, this is the time usually we find when the number of open florets are higher and also the pollen viability is highest. So that is why usually we prefer now that this is the time when we go for this kind of in vivo pollination assay in the field grown plants.
Okay. So I'm sure
Yes.
all of the questions
Yes.
have been answered, and
These are the related and many other questions which is not today's topic, but yes, we will provide you all the questions to the speaker. And this is our end today. It was a great webinar, and it is our
53rd
opportunity. Yes, 53rd, and
wow, it's really nice.
We are very happy that today we at least we bring the bamboos in the Bioingene. It's my target to bring all the plants, all the different plants, and to know their struggle. But yes, it's a very promising, as you included few important topics like in bioenergy. Also, you little give the essence of the tissue culture things which is related, and entrepreneurship for the bamboos, because few crops I know where the everything is economically important, and you can bring anything from the whole plants. Bamboo is one of the such crops where you can use multiple applications, and with this type of research we can explore more applications for this crop. And I congratulate you and your team for such dedicated work for the long time. And it must be tough because when you work in some crops which is very, very long lifespan or long physiological parameters are so long. So it's very difficult to sometimes to get the collection sample and the timing. And thanks for giving us such opportunity to present your work in this platform. And as you know, many international participants are there. So obviously they all are entertained, and some people are also asking questions relevant to their countries. But yes, you have mentioned everything in that way. And we love it. Many people comment on your presentations, your English speaking skill, and your presentation, your slides. So I really enjoyed.
I really want to thank you, Dr. Majumdar and Madam, for such
It was a great pleasure.
conducting in such a nice way. And really, it was well organized. And I was just checking your website. You have so many renowned speakers, and you are basically kind of canvassing for plant science, and that is very important, I think. All of us who are involved in plant science, because nowadays I see this increasing trend that people feel that medical biology is only the biology, but that is not true. Actually, plant science has even more ability to contribute to the world, to live in a sustainable world. And that is why wherever we are, in whatever capacity we have, I think we should be canvassing about plant, and we should be speaking out for more fund and grant for plant science research. So I again thank you. Such a lovely platform you have created. You have such a global audience. I checked the registration list. It is pretty much all over the world. That shows the success of Bioingene. And I know that in such a dedicated way, you two are managing it. It is not easy. It is very difficult, even after managing your personal life, and also to spend some time, also your own lab life, and then to spend so much time to organize this. So I really thank you Thank you both so much for organizing this.
Thanks. It's all about you all, you guys
so
also.
much.
Otherwise, we can't bring such good things. What I enjoyed from Bioingene, that we can know you in a different ways. We know your paper from Google Scholars or anything, but your journey is very important, because we may not working the same field, but our intention is same. Some soft skills are same, some problem are same, and obviously one of those is the funding. But yes, we need to bring that thing, because in other disciplines it's available. In plants, it's also available, like ASPB, American Society of Plant Biology. They are doing that thing.
Yes.
But in Bioingene, we are trying to focus on Asian, African, and Asian-related countries where agriculture is important, and we try to focus the plants which were available here in this continent, not only the model plant Arabidopsis and tobacco. So it's our intention to bring everyone in this platform, and we doing it for the promotions of plant science.
The interview session is particularly popular.
Yes.
In interview session.
That's very unique. Very unique part.
Yes, unique. And when someone asks me to give a talk for 10 minutes, 20 minutes, it's very difficult for me, because when you work in 10 to 12 years on a crops, and there are so many things you want to say. Sure. But that's why we plan for the 2 hours.
It's great.
And majority students, postdoc, and because statistically YouTube, our viewers is 18 to 34 age group.
Oh, nice.
So most of the viewers are there in that particular groups, and it gives us the also motivate us to doing that thing. And we are looking for always the speakers. If you have any connections with any speakers, please let us know, so we can put their talk to. And ultimately, thanks to all the audience for bringing to hear us, and please use this knowledge for your growth, your plant science growth. And yes, one day we'll made a platform that where people are looking for it, that okay,
Yes.
here we can learn something, where you can collaborate, where you can look at genuine lab and genuine papers. Because now it's also very difficult to find out the falsified data and all. So you also need when that, okay, he is a that, and after 3-4 years one paper comes, you can relate somehow that, okay, this is the ultimate result of that experiment and that. So it's a very fascinating. I feel enjoyed in that, and obviously it's sometimes it's challenging to maintain Bioingene, but yes, we will do that. We'll do, and it's always in that way.
The love and appreciation from speakers such as you and the viewers keep us going. That's all it is.
Because it's very hard, like I say, even after managing your personal life, you are spending so much time for the promotion of science, and so that shows your dedication and determination. So I was really intrigued by that.
Sometimes it's give me something share in social media, because I am not preferred to sharing anything in social media. So it's give me something to to run my social media also.
And actually, this is how I first knew you, by Bioingene. And then we came to know each other for some other reasons.
And that is the one benefit. That is the one benefit. When we start, we are not anticipate all the outcome of Bioingene.
Correct.
But now there is so much thing. We can do many more things in Bioingene. Let's see, and obviously your help and your guidance in the future, we love to. And when your students are doing postdoc and good work, we will ask them also to come and give a talk into here. That's why your family and of course is building in that way. Thanks once again. Thank you
to
coming.
Thank you so much.
Thank you.
Thank everyone.
Please come again with more research.
Yes, definitely.
Thank you.
I'll
Also,
make
thanks
sure.
to the viewers.
Okay. Thanks.
Okay,
Soma,
thank you. Bye.
Okay, bye. No.
Bye.
Soma, please stop every.
Okay, I will stop the
live,
stream.
Yes. Live streaming and all. Thanks, everyone.